The current methods of delivering chemotherapy have traditionally been IV, but within the last 2 or 3 years, some centers have moved to delivering the same chemotherapy intraperitoneal. So with a catheter inserted into the abdomen so that the chemotherapy can be delivered directly into the abdomen. And this has proven to be to prolong survival. It is a more toxic approach, and women have more side effects with this type of therapy, but if they can tolerate the side effects, it actually provides a longer-term survival. And there is a movement now to take some of those intraperitoneal therapies and couple it with high temperatures. And when I say high, I don't mean basking in the Bermuda sun. The temperatures are about 42 degrees, so not much higher than body temperatures. But cells become less stable when they move away from normal body temperature. And so even 42 degrees seems to be effective in allowing chemotherapies to more easily destroy those cells than when they are treated at normal body temperature. Avastin is one of the category of drugs called targeted therapies. The targeted therapies come out of the basic or fundamental research that identifies the molecular pathways that are active in contributing to the development of the tumors. And so by identifying the proteins, for example, in a cell that are contributing to the proliferation of those cells, you can then find ways to stop the activity of that protein and hopefully then impair the ability of that cell to survive. And so there's been a number of targeted therapies developed. One of the ones that are currently under investigation is Avastin. Avastin actually targets not the cancer cells themselves, but what are called VEGF, vascular endothelial growth factor receptors that are present on the blood vessels. And so it's not targeting the cancer cells per se, but targeting the blood vessels cancers. And it has shown some promise in a few studies with ovarian cancer patients. In one study, adding Avastin to the chemotherapy mix helped women survive without disease progression for about 2 months longer. So their survival was 18.3 months versus 16 months, 16 months being those who had chemotherapy alone. In another study, when Avastin was used alone compared to chemotherapy, the progression-free survival was improved by about 4 months, 14.1 months versus 10.3 months for progression-free survival. So there is improvement. It's not clearly as dramatic as we would like it to be, but there is rationale for increasing the number of studies that are being done with Avastin on ovarian cancer patients to see whether it might be effective either on its own or in combination with current standard chemotherapy. Probably one of the most exciting new therapies are PARP inhibitors. This is a new brand of targeted therapy based on I'm just trying to look up what PARP stands for because it's a big long name that I don't normally use. Polyadenosine diphosphate ribose. And PARP is an enzyme that's involved in DNA repair. And DNA repair is what's necessary for cells to do every time there's damage. And if you can inhibit the PARP, you will impair the ability for the cell to fix itself if something goes wrong. And this is turning out to have some very promising results in a number of different cancers, but it's particularly relevant to ovarian cancer because of BRCA1. Now, BRCA1 is actually involved in DNA repair as well. And BRCA1 repairs DNA using one method, and PARP repairs DNA using another method. So there's 2 DNA repair methods in the cell, and if you have a BRCA1 mutation that has prevented the one repair method from functioning, and then you can administer a PARP inhibitor to prevent the other DNA repair method from functioning, you essentially hold the cell hostage. It can no way fix itself if there is any damage, and so you actually will kill the cells much more effectively because it has no opportunity to repair any damage. So PARP inhibitors have been tested primarily in women or men who have BRCA1 mutations or BRCA2 mutations. And so in an initial study, there was a number of studies now. I just did a search on the NIH site that has 11 PARP trials either listed as recruiting or in the planning stages. There are none that I could see currently in the National Cancer Institute of Canada website, but I know that there are many being planned. So this is a really exciting new approach. It turns out that the BRCA1 mutation carriers might be most benefited from this. However, it looks like even the non-mutation carriers might be responsive to these inhibitors as well. So this is probably the most exciting targeted therapy in the up-and-coming trials for ovarian cancer. And I'll just highlight again the subtype-specific disease trials. These are, again, focusing on the differences between the different subtypes, serous, endometrioid, mucinous, and clear cell, to be able to design trials that are specific for those different subtypes of cancer. Translational research. This is the category where we're trying to take things from the lab and bring it into the clinic as quickly as possible. There is a large number of groups, both in the U.S. and Canada, looking at early detection, either using blood or urine or even in vaginal secretions. The group in Vancouver is trying to start a study using vaginal secretions to see if there are markers that are indicative of or that can detect early ovarian cancer. And there are a number of these that have been identified. Probably the most promising for ovarian cancer is one called HE4, and its sensitivity and specificity for detecting ovarian cancer seems to be much better than anything else that is available. And it is being now tested in combination with many other blood markers. Probably the most powerful method right now has about 6 markers that are used and are tested simultaneously, and that has a sensitivity of detection of early cancer of about 95%. So we're getting into the stage where by identifying panels of markers that we should be able within the next few years to have panels of markers that can be used for detection of early disease. This field has come a long way since 2002 when the very first study came out on showing that there are proteins that are present in the blood of cancer patients that are not present in women who are healthy. And so although 8 years or 9 years sounds like a long time, this field really has advanced significantly and is very promising. I just want to mention briefly the CUR program. This is the Canadian Ovarian Experimental Unified Resource. It's a pan-Canadian or trans-Canadian study. It involves almost every ovarian cancer researcher in Canada, and particularly those that collect ovarian cancer tissues. And the sole purpose of this resource, this program, is to collect tumors for analysis and identification of biomarkers. Biomarkers that can help segregate the cancers into their different subtypes, and biomarkers that can be used to predict response to various drugs, to various chemotherapy drugs. And the third element of translational research is shown here as next generation sequencing. This is really an exciting time when the technology is allowing us to look at everything that is going on in a cell all at the same time, rather than looking at candidates, rather than looking at, well, gee, I think this gene might be involved, let's just see whether it's different, if it's highly expressed in ovarian cancer. Now we can take cancer and just sequence the whole, what's called the transcriptome, all the things that the genes are making in that cell, and look for mutations. And probably in Canada, David Huntsman, again in Vancouver, is the leader of the pack in this field. He's recently identified, for example, a tumor suppressor that is commonly mutated in clear cell carcinomas. And this is a tumor suppressor that wasn't ever even associated with ovarian cancers before. So now that we know that this tumor suppressor exists, we can go and find if there's ways to understand how it contributes to the development of clear cell carcinomas. And the last slide is on fundamental research. And there's so much of this going on, it's hard to know where to begin, but it all generally lies in investigating what's going on inside the cancer cells that is contributing to their ability to convert from normal to cancer. And then even taking those cancer cells and seeing, are there proteins, are there factors in those cancer cells that we can target, that we can modify their behavior if we target those factors. One example is Dr. Mesmasson's lab has identified a factor called RAN. and they distinguish between the borderline or the low malignant potential tumors and the high-grade cancers. And she finds that if you disrupt RAN activity or RAN expression, then you can make those tumors far less aggressive. So this is a good target for a new targeted treatment. We need to develop inhibitors then for RAN. And the media recently had a whole run on what are called the microRNAs, particularly one called miR-429 that reduces the metastatic behavior of ovarian cancer cells. And so again, it would be great to see if we could stimulate miR-429 activity in ovarian tumors to see whether you could reduce their ability to metastasize further from the ovary. And finally, the last example of fundamental research is just to note that here in Ontario we have what's called the Ontario Institute for Cancer Research, and they have recently developed what they call a selective therapies platform. And again, this is a high throughput system where they're using methods to interfere with different pathways to see which is most effective at causing cell death and cancer cell death. And so this is what's called a high throughput evaluation, a large-scale screening study that will hopefully identify even more pathways that can be targeted for the development of new cancer treatments. And with that, I will finish my presentation, and I guess now we can open it for questions.